So in this session, we'll be taking up the problem summary related to the week 2. So initially, I'll be discussing some summary and sample problems about the week 2 lectures. Then we'll be taking the practice problems for today. Okay, to start with, understanding macromolecules or biological macromolecules, which are macromolecules or building blocks of life. Firstly, the term macromolecules. It tells us that massive molecules form through the polymerization of smaller units called monomers. Initially, this term macromolecules was coined by Hermann Staudinger in 1920, and biological macromolecules are part. So, biological macromolecules are large and complex molecules that are essential for various biological processes in living organisms. We have some types of biological macromolecules. So, four main types are proteins, carbohydrates, lipids, and nucleic acids. So, proteins are fundamental macromolecules with diverse roles in cellular process. They are composed of amino acid chains which fold into specific three-dimensional shapes. These serve as enzymes catalyzing chemical reactions, structural components maintaining cellular integrity, transporters facilitating the movement of molecular movement, and signaling molecules helping in communication within the cell. For example, let's just take a common known protein, hemoglobin, which is responsible for oxygen transport in blood, and other thing is antibodies defending against the infections. Next is carbohydrates, which are crucial macromolecules, function as both energy source and structural support in cells. So these carbohydrates are polymers of carbon, hydrogen, and oxygen exist in various forms. These various forms include monosaccharides, disaccharides, and polysaccharides. So the common known example is glucose, which acts like a primary energy source, while cellulose serves as a structural component in plant cell walls. Next, we have lipids, which are known for their hydrophobic nature and play essential roles in energy storage, insulation, and formation of cell membranes. So we have this phospholipids, which contribute to the structural integrity of cell membranes, and we have cholesterol, which plays a regulatory role in cellular process. Finally, we have nucleic acids. So namely DNA, RNA, vital has nucleic acids. So they are involved in genetic information storage and transmission. So they are structured as polymers of nucleotides. So these nucleic acids consist of a pentose sugar, a phosphate group, and a nitrogenous base. DNA with its double helical structure serves as the primary repository for hereditary information, while RNA, usually single-stranded, acts as a messenger in protein synthesis. Together, these molecules intricately contribute to the structure, function, and regulation of cells. So let's understand how these macromolecules are interconnected, like as we studied the concept of central dogma in the previous week. So nucleic acids store genetic information which guides the synthesis of RNA and proteins. So there are many such ways where proteins are crucial for the synthesis of lipids and carbohydrates. So overall, these four major biological macromolecules such as carbohydrates, nucleic acids, proteins, and lipids collectively contribute to the integrated processes of life. Okay, now I have one small question on week 1 lecture. You can answer this question using the chat box. So who are joined lately? I'm just repeating again. So we have started with the summary to the week 2 lectures. So here I'm showing a question on my screen which tells that which macromolecule stores hereditary information in cells. So here the question tells what is the macromolecule that stores the hereditary information, genetic information in cells. Okay, I see some answers coming up in the chat. Yeah, so everyone can access the chat and see my screen, right? If anyone is facing the problem, please let me know. Okay, yeah, so all the answers in the chat are correct. Thank you for your answers. As I told, nucleic acids, mostly DNA, which store the genetic information, and this information guides the synthesis of RNA and proteins. So now that we know nucleic acids are important for genetic information, I have one more question on nucleic acids. What is the basic structural unit of a nucleic acids? In the previous question, we got to understand that genetic information or hereditary information in cells is stored as nucleic acids. So what is the form or a structural unit, basic structural unit? Yeah, this is nucleotides. So here, in generally, nucleic acids, namely DNA and RNA, are structured as polymers of nucleotides. These nucleic acids overall structure consists of a pentose sugar structure, a phosphate group, and a nitrogenous base. So nucleic acids are the basic structural units of nucleic acids. Okay, now that we know proteins are important, and let's study what are the building blocks of proteins. So those are amino acids. So amino acids are organic compounds containing a basic amino group such as NH2, carboxyl group, COOH. When we see a protein structure or a protein sequence, mainly consists of a long chain of amino acids forming both peptides or proteins. So among the 20 amino acids, some are synthesized by the body and some are not. So the amino acids which our body can synthesize are termed as a non-essential amino acids, such as example alanine, asparagine, arginine, aspartic acid, glutamic acid, cysteine, proline, glycine, serine, and tyrosine are non-essential amino acids. So that means these amino acids are synthesized by our body. And then we have essential amino acids which our body cannot synthesize, must be obtained through diet, such as isoleucine, histidine, lysine, tryptophan, methionine, and some more. So the common structural features of an amino acid include a amino group NH3 carboxylate group. So in the image, the left is the amine group with nitrogen indicated by red, and we have a carboxylate CO group and hydrogen bonded to the same carbon atom. So this central, so this distinguishing the feature is the side chain or the R group. So now that we know the basic structure contains an amino group, carboxylate group, and hydrogen bonded to central C alpha atom. So how come we have different types of amino acids possible? So the different comes in the R group. So that overall each amino acid has 4 groups attached to the central C alpha atom. That is one amino group, one carboxylate group, hydrogen atom, and a side chain R group. So now that we know the R group or the side chain R group decides upon the different types of amino acids, let's look into the classification of different amino acids. So we have properties such as charge, hydrophilicity, hydrophobicity, size, and functional group, which influence the classification. In general, when we see a water-soluble proteins have very hydrophobic residues exposing the hydrophilic side chains. When we come to membrane proteins, which have exposed hydrophobic amino acids, anchoring them into the lipid bilayer. So the different properties or the diverse properties of amino acids are based on their side chain structures. So this Venn diagram shows breakdown of some categories. First, let us take aliphatic. So these amino acids have aliphatic side chains which are linear and unbranched. So example glycine or alanine, valine, leucine, isoleucine and proline. Next, we have some small amino acids. The size of amino acid, whether it's small or large, is based on the relative size comparisons of their side chains. So glycine is the smallest, alanine, serine, threonine, like that. Then we have polar amino acids. Polar amino acids have hydrophilic side chains that can form hydrogen bonds. So they are serine, threonine, cysteine, and tyrosine, like that. Now we have a concept of charged amino acids. So we have two types of charged amino acids based on the charge that is available on their side chain. Positively charged amino acids and negatively charged amino acids. Positively charged amino acids or basic amino acids are lysine, arginine, and histidine. And negatively charged amino acids or acidic amino acids are aspartic acid and glutamic acid. Here we have hydrophobic amino acids. So hydrophobic amino acids have non-polar side chains and are typically repelled by water. And then we have aromatic, which have aromatic ring structures in their side chains, which are phenylalanine, tyrosine, and tryptophan. So understanding these categories helps in predicting the behavior of amino acid in various biological contexts, including their roles in protein structure, function, and interactions. Okay, now that we know some classifications, let's see into some special properties of some amino acids. Cysteine can form covalent disulfide bonds. Proline is in cyclic form. The side chain has been folded into a cyclic structure. Then we know glycine is more flexible than other amino acids, and we have various hydrophobicity scales exist for amino acid residues based on the side chain residues. Yeah, here the convention in this figure just use for green for carbon atom, red for a nitrogen, blue for hydrogen, and pink to denote oxygen. But the major concept to be taken from this is this three: the amine group, the hydrogen atom C alpha, and the carboxylic group are common in all the amino acid structures. The only which varies is the R group. So here I have what sets amino acids apart from each other, whether their ability to do hydrogen bonding or their side chain or R group, amino group, or a carboxylic group. So here I'm showing a question which is related to classification of amino acids. So based on what the amino acids are classified, or what separates one amino acid apart from each other. Okay. Yeah, it's a side chain or a R group of an amino acid that sets one amino acid apart from other. Now that we know different classifications of amino acids is possible, so what amino acids are charged and are basic? So we have learned we have charged amino acids further subgrouped into basic amino acids or acidic amino acids. So what are the amino acids that are charged and are considered basic? The charged amino acids are generally classified if we have charge on their side chain. So here basic amino acids, or in turn we can tell positively charged amino acids. So such as lysine, arginine and histidine. So they contain the positive charge in their side chain, hence they are considered basic amino acid. Further, we have one more category of negatively charged amino acids, such as aspartic acid and glutamic acid. Now that we understand the basic structural unit of a protein, let's go into the different levels of protein structures. Yeah, protein exhibit diverse shape and molecular weights categorized as globular, fibrous, and many others. So, protein shape or the structure is crucial for function, and changes may lead to denaturation. So, how this structure is formed? Amino acid composition or the content of amino acids in a protein influences protein properties, determining whether they are acidic, basic, or non-polar proteins. So, in generally, we have four levels of protein structure, that is primary, secondary, tertiary, and quaternary. At the primary level, proteins are defined by their unique sequence of amino acids, forming a linear chain of forming a linear chain. This sequence, like a biological code, sets the foundation for the subsequent level of structure. So this primary structure is the foundation or the starting point for the next levels of structures. Moving to the secondary level, local folding patterns emerge, creating helices and sheets, alpha helices and beta sheets within the chain. So these folds give the protein more intricate shapes resembling spirals and flat structures. Then we have tertiary level, which involves overall 3D arrangement of entire protein. That is, a complex interplay is dictated by the interactions between the distant amino acids. So this three-dimensional level of a protein can be visualized as the unique 3D shape of the entire nucleus or beaded nucleus, which is essential for protein function. Then we have the final level, which is a quaternary level. This introduces a collaboration kind of nature. Here, multiple protein chains or subunits come together to form a fully functional and multi-subunit complex. Initially, we have discussed hemoglobin, which has its role in oxygen transfer. It has four subunits. That means four tertiary level units come together to form a quaternary structure, working in tandem to transport oxygen. So from primary to quaternary, each level intricately contributes to the protein's overall structure, stability, and function, highlighting the remarkable complexity of these essential biomolecules. So here, understanding protein structure and function provides insights into the dynamic vital roles they play in the complexity of a living organism. So here I have what emerges at the secondary level of a protein structure. Local folding pattern. In general, a peptide, yeah, I'm answering to your question.
Sir,
If we have,
sir, this is not dipeptide, it is a diamino acid. Dipeptide means you should have two peptide units. I don't see one.
yeah, so in general, a peptide is named having 2 to 10 amino acids combination. So here I mean a dipeptide means which is a combination of two. Yeah, so here the secondary level of a protein structure denotes the local folding patterns. So here, apart from different levels of structural organization, we have protein classes based on their structures and functions, with three main classes being globular proteins, fibrous proteins, and membrane proteins. So globular proteins, or typically we call them soluble proteins, many function as enzymes, with their role involved in various cellular processes and often exhibit enzymatic activity. Example like enzymes catalyze specific chemical reactions, which are essential for metabolism, DNA manipulation. DNA manipulation is nothing but replication, repair, and transcription, and post translational modifications. Then we have fibrous proteins, which are often structural and provide support and strength. So they contribute to the structural integrity of tissues. So the major component of connective tissues is collagen. Next, keratin, which we often find it in hair and nails. Then we have other class of proteins called membrane proteins. So these proteins are part of cell membranes, which are present, which span the cell membranes. So they have many functions, like they are used for transport across the cell. Uh, they work for signaling. They work as enzymes. They work for attachments and anything. So understanding the distinct roles and structures of these protein classes is crucial for comprehending the complexity. Yeah, yes. Okay, okay. No issue. So these protein classes is crucial for comprehending the complexity of cellular process and diverse function they perform in living organisms. So now let's look at bioinformatics aspect of sequence-based features on these biological macromolecules again. In first concept is bioinformatics. In bioinformatics, it is sequence alignment. So this sequence alignment is known for unveiling the relationships and functions. Sequence alignment is a crucial technique for arranging DNA, RNA, or protein sequences to know the regions of similarity that indicate their potential function, structure, or evolutionary relationship between sequences. So this sequence alignment process involves representing aligned sequences of nucleotides or amino acids within the matrix, with the gaps introduced between the residues to align identical or similar characteristics in successive columns. So if there is a deletion, insertion, a gap, concept of gap has been introduced in bioinformatics. And there is one more concept that causes a deviation is substitutions. So sequence alignment helps to identify the regions of similarity that may indicate functional similarity. Proteins or nucleic acids with similar sequences often perform similar biological functions. Similarity in sequence, we can tell similarities in 3D structures. So that is the understanding behind homology modeling. Next, by aligning sequences, one can infer evolutionary relationship between different species or genes, or conserved regions in alignment suggest common ancestries. In some other studies, sequence alignments are also used to annotate genes and identify various genetic elements within the genome. This is essential for understanding the functional components of DNA. Sequence alignments extend beyond the biological sequences, encompassing applications like calculating distance caused between strings in natural language or financial data like that. So we have three concepts in a sequence alignment. So this alignment helps to identify the locations of insertions, deletions since the diversions of a common ancestry. Phylogenetic programs leverage these residues to discern relationships between sequences. The probabilities arising from the sequence alignments include. So what can we observe from a sequence alignment? That is, match indicates no change since divergence. Next, mismatch signifies a substitution, that is, one amino acid is replaced by other amino acid, or one nucleotide is replaced by other nucleotide. Next is insertion or deletion, like a particular amino acid is inserted or a particular amino acid is deleted. So generally applications include aligning DNA sequence, aids in assembling larger continuous DNA sequences. This comes concept of NGS alignment, and then protein sequence alignment, like we come to know what are. No, I'm not changing the slide. So in the concept of sequence alignment, first one we'll discuss is pairwise sequence alignment. So the alignment is happening between two sequences at a time. It is termed as pairwise sequence alignment. So this is best for in finding best matching alignments between two query sequences. So these methods include dot matrix, dynamic programming, and word methods. Okay, which will be discussed in further slides. So here, so what is the significance of identifying locations of insertions or deletions in a sequence alignment? So why we need to identify insertions or deletions in a sequence alignment? Yeah, so insertions and deletions here. So what does indels? So here here similarities in sequences can imply similarities in 3D structures. This is what I have explained before. Then when we get a sequence alignment, this helps to identify the locations of insertions or deletions. And this used to understand the relationships between the sequences from their diverged common ancestors. So here, if we are similar, we can tell we have similar structure. But the main thing in identifying insertions or deletions in a sequence alignment is to get the evolutionary relation. So here the answer is D. Similarities give. So as I mentioned, the first concept in pairwise sequence alignment is dot matrix. So which is an qualitative and conceptually straightforward method for aligning sequences, although it can be time consuming for large scale analysis. So this method is particularly for visualizing certain sequence features such as insertions, deletions, repeats or inverted repeats in a sequence alignment. To construct a dot matrix as shown in the image, two sequences are represented along the top row, like along the y-axis or the x-axis of a two-dimensional matrix. And in this matrix, a dot is placed at any position where the characters in the corresponding columns match. This results in a recurrence plot, and the size or the intensity of the dot may vary based on the degree of similarity when characters accommodating conservative substitutions change. So here we see some points are falling along the diagonal of the axis. So in closely related sequences, we see many of these points falling in the diagonal axis. So here, regions along the axis, regions sharing significant similarities appear as the lines of the main diagonal. So similar regions like similarity, like which are aligned similarly, are shown along the main diagonal. So today I'll be demonstrating uh, a Python code-based construction of this dot matrix. So in that we'll be seeing how this alignments is happening for the different sequences, how repeats and inverter repeats are identified. We'll see in the demo session today. So here, given that we have dot matrix approach, what is the primary purpose of it? Yeah, given that dot matrix are used to visualize the regions of similarity. So how in a pairwise aligned sequences of dot matrix are represented? As I explained before, to construct a dot matrix plot, two sequences are represented along the x and y axis of a two-dimensional matrix. So it's not a table, so it's a plot. So plot has x and y axis. So the answer is B, along the x and y axis of a matrix. So next concept other than that is the dynamic programming. So dot plot has several limitations because it is limited by the length of the sequences. Only dot plot is visible if the length of the sequences that are aligned is small, and the visualizing of the results will decrease as we increase the sequences that are going to be aligned. So here we call a dynamic programming. So it has several algorithms which are used for breaking down the optimization problems into simpler overlapping subproblems. So here dynamic programming in bioinformatics is widely employed for sequence alignment than dot plot or dot matrix anything. So this includes Needleman algorithm, Smith algorithm, and BLAST, other things. So first one is Needleman algorithm, which is used for global sequence alignment. So these concepts are just here for introduction. Further, in the next week's sessions, we'll be taking a problem solvings on Needleman, Smith, as well as a demo session on how to use BLAST here. So Needleman provides a global alignment. So global alignment is nothing but aligning the sequences completely, right from starting of one sequence and end. The complete sequence is aligned to check the similarity level. Next comes the Smith-Waterman, which is known as a local sequence alignment method. So this is focuses on finding local similarities. That is it, to find the similar regions. Okay, this is the similar region. This is the similar region in an alignment. So it doesn't involve complete alignment of the given two sequences. Then finally we have BLAST. BLAST stands for Basic Local Alignment Search Tool, which is widely used for comparing biological sequences or finding homologs for a given sequence. What does BLAST do? It searches for the local sequence alignments between a query sequence and a database of sequence. Given that you have a collection of sequences and you try to and one target sequence, you try to find the similar sequences to the target ones in the database given. So BLAST employs heuristic way of algorithms to efficiently find the significant matches. So the purpose of BLAST is to identify homologous sequences in database for a given query. And it also includes word seeding and extension type of methods, and also has a scoring system based on the match, mismatch, and gaps. Whenever you see a BLAST alignment, it gives number of matches, number of mismatches, and other things. So here are some dynamic algorithms that provide optimal solutions for global and local sequence alignments, while tool like so thanks for your question. I'll answer that. So the Needleman is a global alignment method. Smith is a local alignment method. So what happens in global alignment method is, given two sequences, you try to align them completely from start to end. Next comes the Smith-Waterman algorithm, where you check for the local regions of conservation or local regions of similarity. You won't align from start to end. Yeah. So I think I answered this question just now. So what would be the purpose of a Needleman algorithm in bioinformatics? So Needleman algorithm is used for global sequence alignment. Yeah, thank you for your answers. Next, so what algorithm is specifically designed for? Right, Smith Waterman, which is used for local. So we have further sessions in the weeks coming up to solve how to make an alignment using Needleman's method and a Smith method as well. So I hope this is the last question for the summary part. So what does BLAST stand for? So it's expansion of BLAST. Yeah, thank you for your answers. So BLAST stands for Basic Local Alignment Search Tool. Okay, so now that we have completed the summary sessions, so before moving on to the actual quiz session, I would like to demonstrate a construction of a dot matrix. So is so I'm using a Google collaboratory Python code here so that it will be easy for me to share with you later on. So I hope everyone can see my screen clearly. Yeah, thank you for letting me know. So here initially I'm making some Python functions. As we know, dot matrix require input of two sequences. So this function will take sequence 1 and sequence 2 as inputs. Further, I used different libraries of Python programming to make the matrix look good. So for this example purpose, I have taken the two sequences. So kindly have a close look at these two sequences. So here for our initial understanding, I have taken very similar sequences, and that is simple sequences, which are very like similar sequences, almost same. Then comes the dot matrix. So sequence 1 have taken on y-axis, sequence 2 have taken on x-axis. Now even in sequence 1, if we have A, we have placed a dot here. Near to the origin in the leftmost corner. So based on the similarity, so if amino acid is present on x-axis and a similar amino acid is present on y-axis, we keep on placing the dots. Since this is completely related sequence, we see all the dots are aligned along the diagonal here, very well along the diagonal. So given the sequence here, the sequence has some repeats, that is ACGT, ACGT, ACGT is completely repeated here. So these repeats are indicated along the parallel lines that form across. So this is one parallel line. This is the next parallel line. So these parallel lines indicate repeats in a dot matrix. So here I have taken some little complex English terminology based input. I'll show the input. I have changed to some English sentences. The quick brown fox jumps over the lazy brown dog. Here have changed this jumps to an inverted order. So let's see how this inverted segment of sequence is identified in a. Now that I know I wantedly change this, now let's visualize this inverted in a dot matrix. So given that dot matrix is here. So this is the dot matrix, again sequence 1 on y axis and sequence 2 on x axis. Since the both sentences are almost similar, we see a thick line along the diagonal, and here in between we see something which is perpendicular to the diagonal. This tells about the inverted repeats. Parallel lines tell about the direct repeats. This tells about the inverted repeats. Yeah, yes, please. Yeah, someone has raised the hand. Yes, please.
Sir, is there any rule that sequence 1 should come in x-axis, y-axis, and sequence 2 in y-axis? Shall
No,
we, uh, what? Shall
yeah,
we change that, sir?
yeah, I
Sequence 2
understand
in.
your question. Yeah, I understand
Uh,
your question. Yeah, thank you for this raising this question. There is no such limitations in dot matrix or in any other alignment methods. But if in further also, we'll be taking up on the dynamic programming methods where I told Needleman method, uh, Smith method. There is no such rule.
yes.
The axis has to
Okay.
be in particular direction.
Okay.
Based on the
Okay.
axis, the alignment will change how you take, but the information what we gain won't change.
Okay, sir. Thank you.
Yeah, so this is a small demonstration. So along with the PPT slides, I'll also share this for your practice purpose. Now, is this demonstration clear? Thank you. Yeah, this is just a simple Google Colab file, where in the Google you can type Google Colab, then open this file. You will see the similar interface, and then you can work on this code. And you can change your input sequences however you want for your understanding purpose. Here I have taken smaller sequences so that it will be easy to explain. Okay, now I'll be moving on to the assignment quiz. So today we have most of theory questions only, so that should be fast. So which or what is the one element that proteins have but carbohydrates and lipids do not have? Yeah, so along with the answers, can anyone mention in what form proteins have the missing one? What is the compound? Yeah, yeah. So we have learned proteins, carbohydrates, lipids, and nucleic acids as the biological macromolecules, so which are made up of carbon, nitrogen, oxygen, and hydrogen. But still, they have their internal differences in having these bio compounds, biological atoms. So here, so proteins have nitrogen, but carbohydrates and lipids do not have. Let's look into this some structural image. So in terms of carbohydrates, we see only the carbon group, we see hydrogen group and the oxygen group. In proteins, we have amine group NH3, and in some side chains also we have amine groups to have nitrogen atom. In nucleic acids, we have a nitrogenous base itself which contains a nitrogen group, and in lipids, we don't have any nitrogen. So the element which is present in proteins and nucleic acids and that is absent in carbohydrates and lipids is nitrogen. So given that protein structure exists in 4 levels, what level of protein structure is influenced primarily by hydrogen bonding? Okay, different answer. Yeah, it's the secondary structure. So the major driving forces for the forming secondary structure is the hydrogen bonding. So the formation of tertiary structure has several other driving factors other than this hydrogen bondings. So there we comes to a concept of protein stability. There we will be studying about hydrophobic effect, where burying of hydrophobic residues away from the protein surface or water. So have you seen here the two known secondary structures of protein are alpha helix and beta pleated sheets, where the alpha helix are stabilized by the hydrogen bonding to maintain their alpha helical structure here, and beta sheets have hydrogen bonds to maintain their beta pleated shape. So these hydrogen bonds provide stability to the secondary structures, thereby further allowing them to fold into tertiary structures. Next is that, so hemoglobin is tend to have 4 identical subunits. So what is that level of structure known as? Yeah, if anyone is unable to follow the questions, please let me know if I move fast so that I'll explain again. Even the recording of this lecture will be available to you by tomorrow morning. Yeah. So here, having 4 identical subunits is kind of having that hemoglobin bundle. So the quaternary structure, the final level, which is a functional form of a hemoglobin. So what is the function of hemoglobin? Major function? Yeah, the major function is to transport oxygen. Thank you for your answer. So this is the image showing the overall levels of protein structure. So in the quaternary structure, the protein consists of more than one amino acid chain. So here I have given a disease cell composition in a disease state. So first one is normal hemoglobin. Second one is a hemoglobin in the patient of sickle cell anemia. So when we consider these two, like we have a normal hemoglobin and a sickle cell hemoglobin, and I have made two questions to answer the both. So let's take the first one. Given two hemoglobins, which type of hemoglobin is apparently more polar and soluble in water? Kindly observe the images for what is the change. So in normal hemoglobin, we have glutamic acid, whereas when this glutamic acid is mutated to valine, we lead into a condition called sickle cell anemia. Only three answers. So can I take others for sickle cell anemia as the answer? I see three answers as normal. Yeah, so in the first case, thank you, Anupam, is a normal hemoglobin because presence of the polar amino acid. In second case, this polar has been mutated to valine. What happens? So which type of hemoglobin is more nonpolar and insoluble? So now we are taking the second one. Which type of hemoglobin of given normal and sickle cell is more nonpolar and insoluble? Yeah. So the normal hemoglobin, its charged amino acid is replaced by an hydrophobic amino acid, mutated to or substituted. So this makes this particular sequence more nonpolar and insoluble because of a presence of a nonpolar group. So here we have sickle cell hemoglobin. So moving on to the fifth one. Proteins which are embedded in the outer membrane are known as yeah. So generally membrane proteins are known to interact with the lipid bilayer either by adopting alpha helical or a beta barrel structural fold. So one class of membrane proteins is a beta barrel membrane proteins, which are mostly observed in outer membrane proteins. So these membrane proteins have a characteristic structure resembling a beta barrel. So beta sheets fold themselves to form a barrel kind of structure. So here, this is how a schematic diagram for a beta barrel protein looks like. Okay, next, given that we had a little understanding about membrane proteins. So one class of membrane proteins is a channel proteins. So what is the main purpose of the channel proteins? Yeah. So channel proteins are type of integral membrane proteins that facilitate the movement of specific ions or molecules across the biological membrane. Typically, this biological membrane means cell membrane. So these protein forms channels or pores that allow selective and controlled passage of substances, maintaining the integrity of cellular compartments and enabling various physiological processes. So the function of a channel protein is gating and selective ion conduction. Yeah, yes, please.
Sir, what does comprehensive database mean?
It's a generally comprehensive database or whatever is it. It has information about everything, complete. So it is for an enzyme database. Brenda is known for enzyme database, and it has comprehensive information about everything.
Sir, I didn't get you, sir. Your voice was breaking.
Sorry. So here the comprehensive name is added to a database if it has information particularly about one thing.
Okay, sir.
Yeah. So given the enzyme nomenclature, so we have 6 classes. So these classes means the general type of reaction the enzyme catalyzes. The first one is oxidoreductases, which catalyze the oxidation reduction reactions. Next is the transferases, which are involved in transfer of functional groups. Next we have hydrolases, which catalyze the hydrolysis reactions. Next, lyases, which help in the removal of groups without hydrolysis. Next we have isomerases and ligases. The last class is ligase, which catalyze the joining of two molecules using an energy derived from ATP. Next, we have a concept of matrices. So the first one is PAM matrix. These are just introduction question for a PAM matrix. So meanwhile you answer, I'll be giving a brief intro to a PAM. So a PAM matrix, which stands for point accepted mutation matrix is a tool used in bioinformatics and computational biology to quantify the evolutionary distance between two homologous proteins or DNA sequences. It helps in assessing the likelihood of mutations occurring at a specific position within these sequences. The concept of PAM matrix was introduced by Margaret Dayhoff in 1970s. The basic idea behind a PAM model is to quantify the probability of particular amino acid substitution over the time, and it also considers constant rate of evolution. Here we try to find the probability of a particular amino acid substitution or a mutation. So first thing is constructed based on the observed substitutions in an closely related sequences. So this observation of substitutions in closely related sequences represents the probability of one amino acid changing to other over a short evolutionary period. Then we have different versions of PAM like PAM1, PAM20, PAM250, PAM500, etc. So each PAM matrix is intended to represent a specific evolutionary distance or time substitution values. So whatever PAM matrix we take, the values in a PAM matrix indicate probability of a particular amino acid at one position mutating to other amino acid over the specified evolutionary distance. Higher values indicate higher likelihood of substitutions, while lower values suggest less likelihood. So how to select appropriate matrix given that we have many matrices? The choice of the PAM level is often determined by the researcher's knowledge on the evolutionary relationship among the compared sequences. So here the PAM is 1 PAM means accepted point mutation per 100 residues that have been observed. Yeah, here I have added some explanation since this is the first time we have taken. So as mentioned in the last line of this slide, so the distance of the PAM matrix probability value is expressed as number of expected amino acid substitutions per 100 residues in the sequence. So 100 means 10 square. You just have twisted it to 10 square. So the central hub for collection of functional information on proteins with accurate, consistent and rich annotation is it's UniProt. So we have few more alignment questions. After that I'll be demonstrating a UniProt, how to extract information from UniProt. Yeah, I have one more question. UniProt KB is made up of two large sequence collections. One is manually annotated and reviewed, other is automatically annotated and not reviewed. Nice. Yeah, it is SwissProt and TRMBL. SwissProt is manually annotated and reviewed, and TRMBL is the automatically annotated and not reviewed sequences. So I have made an pairwise sequence alignment here and I have provided some scores. a score of match is 3, a mismatch is -1, and a gap. Gap is indicated by a hyphen symbol. So gap is -1. So what could be the net score of this alignment? Yeah, I'll give some time to answer this. Okay, I've got two answers, one for A and one for B. Okay, let's solve this together. So here I have mentioned the sequence. So in the first case, so let's consider like this. In the first case, it is a match. So we have a score of 3. In second case, we have a mismatch. So we have a score of -1. In third case, we have a condition of match and score of 3. And when we introduce a gap, we have a penalty of -1 again. And similarly, mismatch -1, introduction of gap, so gap penalty -1, and again a match 3. So overall it is 9 minus 4 equals to 5. Now, anyone having doubt in this? Okay. To further practice on similar kind, I have one more. The answer is 5. I have one more alignment. So I little change the alignment and the values as well. So try to solve this. And there is a no rule that match score has to be this, mismatch score has to be this, and gap penalty has to be this. Based on your work, based on your analysis, the cutoffs can be decided upon you. Yeah, all of your answers are same. Okay, I'll solve this now. So here we have a situation of match 4, introduction of cap penalty of -2, match 4, introduction of gap penalty of -2, mismatch -1, match 4 again. So here I have given some task. Hope some of you might have tried the task that I have given previously. So for the ones who doesn't know the UniProt, so I'll be having a little basic demonstration on UniProt now. So what I do is go to our browser, whatever the browser, we type uniprot.org, then this will redirect to our UniProt homepage. So if someone is very interested to know about the statistics of UniProt, here we have a statistic option to view. I think yeah. So in the right top corner, we have an option to view statistics of a UniProt database. Then we can see a search page. So in the search page, for example, let me use insulin. I'm using their example. Then I'm clicking on search. So this gives me how to view either as tables or cards. I'm selecting the view as tables. I can see different entries that are related to insulin. So let's shift our focus to the left side tab. First thing on the left side tab we have is status. Under status we have reviewed and unreviewed, which are part of SwissProt. So we have a question on this. So SwissProt consists of manually reviewed entries. This is automatically reviewed. So the overall results, we have 1,36,566 results that have obtained for our search insulin. Under that 5,200, near to 5,200 are reviewed and rest are unreviewed. Next has organism. How many entries of this insulin are corresponding to rat, corresponding to human, and corresponding to mouse, bovine, zebrafish, and etc. we have here. Then we come to know with their extra information, proteins with steady structure. So our search has proteins, 1292 proteins with 3D structure available. This is experimental 3D structure available. Then active site information known, activity regulation known, all other extra information has been given here. So now let me select the first one, insulin receptor. This is of P06213 to view what all the information are given for the each entry of insulin. So the basic information has been given: what is the protein, gene, number of amino acids, and organism, and other details. So if you focus again on the left side, so we have information on function, we have information on names and taxonomy, we have subcellular location, diseases, and other structure and family domains. So here I'd like to directly move to the structure part. Yeah, here you can visualize the three-dimensional structure of the insulin receptor, and we can also see different PDB IDs corresponding to this insulin receptor. Here I can see some X-ray based PDB entries, NMR based also we have on 2F, 2MFR, and when you come to the end of this list, you identify alpha fold related entry. Then this gives the secondary structure information about this insulin receptor, followed by that. Then also it gives integration to other database where data is available on the similar insulin receptor. So it gives information about family, so that it gives what is the insulin binding region, what is the disordered region, and it gives what is the keywords that are corresponding with this. So it's a transmembrane helix. Here we can see the complete sequence information. So it has 1382 amino acids with a molecular weight of 1,56,333 daltons. And when this has been last updated, year of upgradation, here it also provides us to download the sequence. So by clicking on the download, okay, I'll show again. So when we come to the sequence section, here you can see download and some tools like compute molecular weight or peptide mass, prot param, which gives the protein parameters. Followed by that, we have a tab of download. On clicking that, you can extract the FASTA sequence of this protein insulin receptor. Finally, you have similar proteins. What are the similar proteins that are identified? So with 100% identity, we have one. With 90% identity, we have set of proteins. This is kind of similar to a blast result, and with 50% similar identity, what we have. Further, apart from these information, we also have variant viewer, feature viewer, genomic coordinates, and publication. How this structure, how this domain information, binding site information, various literature that has been related to this insulin receptor will be provided here. Here one can also find the external links which similar protein has been the same protein has been reported. For example, we have studied Brenda, which is an enzyme related. So this entry has been made in Brenda as well. So let's try to click on Brenda. I hope everyone can see Brenda. So I'm just navigating from here to Brenda. So this provides the enzyme information. That is, I just zoom. Yeah. Here one can see the enzyme classification tree. So we have studied there is 6 classes for enzymes. So this insulin receptor belongs to transferase class. In that we have further subclasses like transferring phosphorus containing groups. Further, this is the enzyme classification information related to our entry. Interpro can be used to extract the domain related information for this entry. Yeah. So this is what I planned. Yeah. So this is what